Hello, everybody, and welcome to our weekly Extracellular Vesicle Journal Club. Today we have Raluca Ghebosu with us. Hello, Raluca. You're mute.
Sorry. Hi, everyone.
Hello, thank you for joining us. So I will introduce briefly. Raluca is a PhD candidate in Australian Institute of Bioengineering and Nanotechnology at the University of Queensland, Australia. And he recently published a very interesting paper, I think it was August or September, when you published this
Yes.
paper in the journal Extracellular Vesicle. And we are going to talk, you are going to talk about something very interesting, very actual, very actual. You are going to present a new essay to demonstrate and somehow also quantify how, as a semi-quantitative method, how extracellular vesicle interact with lipoprotein. And this assay, this is a potential assay that can be probably found in future, near future commercialization, can be implication in diagnostic, therapeutic, and also basic research application. So now I give all the share to you, and please, you can start. Thank you again.
Thank you so much. Amazing. Perfect. So it's all working. We can all see my screen.
Yes.
Thank you very much, Marco, for the invitation and for everyone for joining me today to talk about, as Marco mentioned, our new work about Lipoprotein Association Fluorometry, which we've coined the LAF assay. I know I got an introduction from Marco, but my name is Raluca and I am based at the University of Queensland in the sunny Brisbane, Australia. Although, as I say that, it is currently raining quite a lot outside, but it is mostly sunny here. So yes, today I'll be talking about our recent publication relating to lipoprotein and extracellular vesicle binding. So I know that this is an EV journal club, but for those of us that are new to the field or just simply joining, I wanted to give a really quick overview about extracellular vesicles just to set the scene. So extracellular vesicles are these unilamellar or multilamellar lipid bilayer-based vesicles. They're released by all cells. They contain biomolecules. They're involved in cell-to-cell communication. And in the past 10 to 15 years, they've really emerged as promising avenues for drug delivery and therapeutic-based systems. So they are really something that has taken the kind of field by storm, but they are also being explored as trying to understand a lot of pathophysiological diseases. So whether that's cancer, neurobiology, diabetic kidney disease, many other different diseases, and we're all starting to try and understand how extracellular vesicles are contributing to biology. The other star of the story that I'll be telling you a bit more today about is lipoproteins. So commonly lipoproteins are known of in terms of good and bad cholesterol. So when you go to the doctor and they say things like you have high cholesterol, they're referring to lipoproteins. Lipoproteins exist in four main categories. That is high density lipoprotein. They're the smallest and the highest density. And they're commonly the good cholesterol that, the doctors want you to have a little bit more of. And then you have low density, very low density, and chylomicrons that again start increasing in size and they fall into the disease pathogenic bad cholesterol. But because they overlap in size and in density to our extracellular vesicles and other biologics, conventional techniques really struggle to separate them. So one of the big challenges in the field, and it'll come up all the time, is how do we separate lipoproteins from our extracellular vesicles? That's one of the questions that will always come up. So I'm going to here to try and flip that a little bit on its head and talk a bit about how we should be looking at the interactions between extracellular vesicles and lipoproteins rather than thinking of them as just merely being a bit of a contaminant and a nuisance in our extracellular vesicle samples. But so because of this and because of the struggles we have in separating them, this highlights that we really have a need for more effective methods to measure lipoprotein binding, especially for extracellular vesicle, nanoparticle, and large biological-based systems. We've seen in human biofluids that there are interactions happening between extracellular vesicles and lipoproteins. So what we have here is, let me just put the laser on, cryogenic electron microscopy samples of just crude human plasma. So you can see here this dark bilayered structures. These are our EVs, whereas here with the single structure and a bit more of sharper edges, these are our lipoproteins. So these interactions are happening just in biofluid, in vivo, in crude human plasma. So they are spontaneously happening all the time. And so that indicated to us that they must be a bit of an important part of extracellular vesicle biology and function. But not just that, when we were looking at in vitro mechanistic studies, we found that if we add LDL, so one type of lipoprotein with EVs, it changes the cellular uptake of EVs. It changes the cytokine released by these cells. So these lipoproteins and EVs have been shown to be able to work together in order to mediate cell function and outcome. And this is just one example. There's a fair few in the literature exploring how extracellular vesicles and lipoproteins might be working together. But as I alluded to before, there are some techniques with which you can assess lipoprotein binding to small molecules predominantly. And those involve things like centrifugation, ultrafiltration, or dialysis. Or on the opposite hand, things like mass spectroscopy or liquid chromatography. However, a lot of these systems are very time costly. They require complex detection equipment, specialist knowledge, and they're also not readily applicable to things like our extracellular vesicles, large biologics, and large nanoparticles. They also need to be specifically tailored to each drug. So they can be quite labor intensive and quite time consuming. And so this is why we thought we have an urgent need to develop a simple and effective high throughput method to quantify extracellular vesicle and lipoprotein interactions, because then if we can do that, then we can start to probe extracellular vesicle biology specifically in relation to lipoproteins. And so this is kind of where the LAF assay, as we've come to call it, came about. So early on in the project, we discovered that very low density lipoprotein can form these visible aggregates in a temperature and concentration dependent manner. So we could see them just under standard light microscopy that was forming, especially at those high concentrations. And we saw that we could measure this change in a fluorescent way. So when we had labeled them with an indocarbazine dye, we saw an increase in fluorescence intensity when we started to, that correlated with when we were seeing these larger visible aggregates. So we wanted to then confirm that what that signal was measuring was due to the lipoprotein itself. So we performed some immunofluorescence staining to show that a key protein of our VLDL, that is ApoB, co-localized with that lipophilic dye. So you can see this is our lipophilic dye, and in the green we have that protein component of the lipoprotein, and they were merging. So these larger fluorescent aggregates were due to the lipoproteins and not just free aggregates of the dye that we were using. We also used a polymer known as polyethylene glycol that is known to precipitate nanoparticles out of solution to further show that this was mirrored by aggregation, or this increase in these visible aggregates. So when we added this polyethylene glycol, an increase in concentrations, we saw more and more of these visible aggregates, and this was correlated to an increase in that fluorescence intensity. So there was a structural change happening in our lipoprotein particles that was interacting with the dye and causing this change in fluorescence intensity. We also just confirmed that it wasn't due to interactions between the polymer and the dye, because it's very important to ensure that there's not something else happening in the background. We could see that adding that polymer didn't change the way that the dye was behaving in the absence of the lipoprotein itself. And so we identified that our kind of method can fluorometrically identify aggregation-associated changes in our lipoprotein particles, so changes that are happening to the molecule due to those large aggregates forming. And so this was kind of what inspired the concept of our LAF assay, because we saw that when there is those structural changes, we have changes in how the dye is partitioning into our lipoprotein. However, we hypothesized that if we added test agents to that mix, so things like our extracellular vesicles, nanoparticles, and things that have been really difficult to probe, they would outcompete the dye for binding, and they would change the structure such that we could see that as a semi-quantitative change in fluorescence. So if our hypothesis was that if these test agents was to bind to our lipoprotein, that would cause a decrease in the fluorescence intensity. And so that was exactly what we set out to try and see if we could measure that. And so we created our assay, which this is the standard workflow. As you can see, we just add our 3 reagents, our lipoprotein of choice, our test agents, and our fluorescent molecule to a plate, incubate it for 1 hour, and then measure it on a plate reader quantitatively. So it is a really simple workflow, especially compared to some of those larger, more time-consuming methods that I described earlier. The first thing we wanted to do was to assess small molecules. So these are clinically approved small molecules that are known to be extensively bound to plasma proteins. And so we picked warfarin because we know it is about 99% bound to plasma proteins in the blood because it is used widely in clinic as an anticoagulant. And what we found in our assay was that there was this significant decrease in fluorescence intensity, indicating that there was binding happening between our warfarin and our VLDL. We also saw this in a concentration-dependent manner. So when we added more and more of our warfarin, we saw a greater decrease in fluorescence intensity, indicating to us that there was more binding of this drug happening to our lipoproteins. We also looked at this under the standard light microscope, as we did earlier, and you can visibly see changes in those aggregates that are forming. We also tested it with another clinically approved small molecule, as well as a non-clinically approved small molecule as well, known as cytochalasin D, so it's commonly used, to see that we can see different levels in plasma protein binding based on the small molecules that is using. So we determined that when we add these small molecules, that we know that they should bind to lipoproteins, we can measure changes in that self-association, which we see as visible large aggregates. So what we wanted to do next was to start probing what we were really interested in. And these were larger based nanoparticles, things that have been really difficult to probe in the past. So we started first with synthetic based nanoparticles because they have been shown in the literature to bind to lipoproteins. So we thought that they would be a really great starting point for us. And we also used nanoparticles that had this PEG shell coating. And this, conversely to the opposite PEG I mentioned earlier, the polymer form, this PEG is known to have a stealth effect. So it's actually meant to prevent the binding to the lipoproteins. And what we saw was indeed this green nanoparticle here, which we know that should bind to lipoproteins based on the literature, did have a significant binding. And we saw that the ones that we expected to not also did not. And there was a significant difference between the two that we could measure. We also tested a lipoprotein interacting polymer and found that again, that did bind. So next we went to go to our EVs, which is what we were really interested in finding out and trying to probe extracellular vesicle biology. So I tested extracellular vesicles from a wide range of human and non-human sources and found that they did all in fact bind to varying extents to lipoproteins. So I think orange-derived EVs were the only ones that didn't show any binding. The rest did in fact show binding to our VLDL. And so what we wanted to do next and take a next step is, okay, great, we've shown that this assay can be applicable to extracellular vesicles, biologics, nanoparticles, small molecules. What about trying to understand disease pathologies? And so we isolated extracellular vesicles from primary human natural killer cells and compared them to cancerous natural killer cell derived EVs and found that actually those from the cancerous NK cells had significantly more binding to VLDL than those that we derived from our healthy NK cells. So we saw that there was this disease effect happening dependent on whether the cells were cancerous or not. So we wanted to then explore that phenotype a little bit more and uncover, okay, we have a difference between cancerous and non-cancerous, but what about disease progression? What about metastases? So we then set to isolate extracellular vesicles from a wide range of triple-negative breast cancer cell lines. And I didn't show it for the previous EVs, but it is all there in the publication. I just didn't want to show too much of the same thing. But all of the extracellular vesicles we isolated, they were validated and authenticated using nanoparticle tracking analysis, Western blot, where we showed the enrichment of extracellular vesicle markers, CD63, CD81, ALEX, and then the absence of contaminant markers such as calnexin. And we also validated the bilayer appearance through cryogenic transmission electron microscopy. So we did this and authenticated all of our EVs, and the more extensive authentication is available in the publication on the Journal of Extracellular Vesicles. But these extracellular vesicles that we're going to talk about now, they're coming from a triple-negative breast cancer cell line, mostly the ones in blue. The one in green are estrogen receptor breast cancer. So they will be our poorly metastatic control, whereas all the ones in the blue are our highly metastatic control. And what we found was that the extracellular vesicles derived from highly metastatic triple-negative breast cancer actually had significantly more binding to our lipoproteins than those that were from poorly metastatic breast cancer-derived cells. And not only that, we performed this in triplicates across 3 separate days to show that it was reproducible across multiple instances, and there was very little variation in the data and the trends that we saw. So what we were able to find was that these metastatic cancer-derived EVs actually interacted more with our VLDL, specifically in this case. And so not only are we seeing a cancer-dependent effect, but we're also seeing a metastatic effect. What better way than to correlate this data than to see it through cryogenic transmission electron microscopy? So we wanted to validate this semi-quantitative data using cryo-EM. What we did was we tallied, for lack of a better word, measured all the instances of binding that we could see in our cryo-TM and found in the top corner here that they correlated with the results that we got in our LAF assay. These healthy derived EVs showed the lowest amount of EVs fusing to VLDL under cryo-EM, followed by our poorly metastatic and then our highly metastatic. And so what you can see here, and we'll go through it a little bit because it's can be a bit difficult to see, especially on Zoom, is these darker structures here. So you can see how some of the structures are quite opaque and filled in. These are our lipoproteins, whereas the structures that we see with these bilayers and a much lighter core, those are extracellular vesicles. And on multiple occasions, you can see that these extracellular vesicles are binding to lipoproteins. Sometimes we saw multiple extracellular vesicles binding to a single lipoprotein, or even a single or multiple lipoproteins, VLDL specifically, binding to a single extracellular vesicles. We characterize this as 2 different types of associations, and that was the direct fusion that you can see happening here between 2 particles, but also surface-based associations, because there could be multiple different ways with which the extracellular vesicles and the lipoproteins are interacting with each other. So they could just be interacting through surface-based receptors that are causing them to bind onto each other. But so cryogenically, through microscopy, we could see that the results we got in our LAF assay correlated to what we could see under the TEM. We didn't want to stop with just a breast cancer model. We then wanted to expand it to a different cancer type to see, well, is this just a breast cancer phenomenon? So what we used was an osteosarcoma-based model. So previously, we had a poorly metastatic and a highly metastatic triple-negative breast cancer that came from different cell lines, different patients. What we used next was a metastatic and non-metastatic system that they both came from the same original cell line. The only difference was the introduction of a KRAS mutation that increased the metastatic potential of this next 143B cell line, as it was called. We also looked at the fluorescence intensity of just the EVs labeled with our dye, and you can see that it was quite low compared to the VLDL in this case. But what we found here was that, again, similar to the breast cancer, we saw a metastatic dependent decrease in fluorescence intensity, which is indicative of more interactions happening between the metastatic osteosarcoma-derived EVs compared to those from non-metastatic cells. So these are match-patient cells. It's just the metastatic potential that's somehow changing these EVs and causing greater interactions with our VLDL. And again, we also validated it through cryo-TEM, where we saw the same trend. These metastatic cancer-derived EVs had more binding to the lipoproteins. So here we could see that what we were seeing under cryo-TEM also was correlating with what we were measuring semi-quantitatively through the LAF assay. So this showed us that these increased interactions from the metastatic cancer EVs with our lipoproteins are present in various cancer types, not just in a breast cancer model, but also in an osteosarcoma model. And we do see pathological differences in even natural killer, which are an immune cell derived EV. But then we thought, okay, well, VLDL is just one type of lipoprotein. What is the potential of this technology to be expanded to other types of lipoproteins? So what we then sought to do was to look at low density lipoprotein, which is a different type. It has higher density than very low density, and a bit of a smaller size. And we saw again that we could measure changes in the structure and association and the partitioning of the dye in this LDL, so we could see changes. And then we added our test agents back in. If you remember, it's been a little bit back now, but warfarin was our positive control. It was the one that we know from the literature as a small molecule should have the highest binding. And we can see that here with our LDL, it did. And also a lot of our EVs, they did show binding to LDL. We didn't see the same metastatic cancer phenotype. However, the cancer-derived EVs did show significantly more binding to LDL than our healthier, I say healthier, it's an immortalized cell line, HEK-derived, HEK293T, which are a human embryonic kidney cell-derived EVs. We also then wanted to test high-density lipoprotein and complete the trifecta. So we could again measure those partitionings, but this time we didn't see as broad changes. And in fact, paradoxically, our metastatic triple-negative breast cancer actually showed an increase in fluorescence intensity, which could be due to just changes and temporary changes or destabilization that might be happening when we're adding in these EVs. And again, though, our positive control, which is the warfarin, and we also added an additional positive control of LPS, that is lipopolysaccharide, that is known in the literature to bind to HDL, and they both show significant binding to high-density lipoprotein. We then we showed that the LAF assay can be expanded and used to test individual types of lipoproteins, whether that's very low density, or high density. And then we got the idea of, well, what about a physiological relevant mix? Because we want to understand how they are working within our bodies. What better source than crude human plasma? So we took human plasma, just crude as it is, and applied the same concept and the same principle as we did in the LAF assay, where we just added, instead of adding a lipoprotein this time, we added a 20% concentration of crude plasma and added in our EVs to see if we could measure changes. And in our positive control, you can see that again, there was quite extensive binding of the warfarin happening to plasma proteins, as we know it should, 99% bound. And again, we saw that there was this significant decrease in fluorescence intensity, which is indicative of binding, with our metastatic triple-negative breast cancer-derived EVs. And this was seen across all donors. So we had different blood types, different sexes, and different ages of all of our donors. And across all of them, these metastatic triple-negative breast cancer EVs were binding to lipoproteins that are present in plasma at physiologically relevant levels. You could also see that there was some differences in between the donors and some different donor variations. So not only could we use this technology to probe individual lipoprotein interactions with extracellular vesicles, nanoparticles, biologics, and small molecules, but we could also apply it to physiologically relevant biofluids, such as human plasma, that contain a physiologically relevant mixture and concentration of lipoproteins, because all 3, 4, 5, 6 lipoproteins, as there are, will be present in human plasma. And so we've shown this broad applicability of the LAF assay. And now what we're really excited is to try and explore in the future, well, how this could change our understanding of extracellular vesicle biology. We have a couple of ideas of how they might be interacting with our extracellular vesicles. So we know that lipoproteins and lipoprotein components can change the surface corona. So that is the kind of protein shell around our extracellular vesicles. So perhaps lipoprotein components are being integrated into the protein biomolecular, corona of these extracellular vesicles, which is affecting things like immune activation, biodistribution and clearance, cell signaling, and even organotropism targeting. So if the biomolecular corona can be altering all of these effects, perhaps this is one mechanism by which lipoproteins are interacting with our extracellular vesicles and mediating their function. Another mechanism is through co-transport. So there has been a study published that showed that complexes of extracellular vesicles and lipoproteins can be transported across cell barriers and remain intact. And so we know that lipoprotein receptors are present in all tissues throughout the bodies. And so we propose that potentially these complexes could mediate cavernous transcytosis across the endothelial barrier and release extracellular vesicles into the vasculature space. So from that tumor microenvironment, help them cross into different tissues and make it, as we saw before, targeting and biodistribution of EVs all throughout the body. But that's what we hope to do. What we've shown so far with our LAP assay is that it is capable of semi-quantitatively measuring lipoprotein binding with a range of different molecules. We've uncovered that there is binding of small molecules, proteins, synthetic nanoparticles, polymers, human, non-human derived EVs to different types of lipoproteins. We've also found that there was distinct binding patterns that were based on the disease state and the metastatic potential of the originating cell. We also then, at the end, applied this principle in this LAF assay to relevant human biofluids that had complex mixtures of lipoproteins. So as I mentioned before, we weren't probing them on a one-to-one basis, but we were looking at the broader picture of a whole. And so now, hopefully, people around the world and us can use the LAF assay to uncover how EV and lipoprotein interactions are contributing to pathophysiological states, but also help us revolutionize drug discovery pipelines for EVs and nanoparticle-based therapeutics, because we need to be able to understand these interactions in order to get the best therapeutic effect of any future extracellular vesicle drug delivery systems, which I know that people around the world are working on, and perhaps some of you in the audience as well. We've also been able to patent this technology, so it is available for licensing, and please reach out to myself or to Adam at the number at the email here as well, if it is something that you're interested in. But on that note, I wanted to thank all of the people in our team, all of the co-work, all of the co-authors, and all of the funding bodies that made not just this work, but all of the work that we do in the lab possible. And please let me know if you have any questions, and thank you very much.
Thank you very much, Raluca. That was a very interesting presentation and very amazing data, especially the CRY-AM data. Let's see if we have any question from the audience. This is time to ask if you have any curiosity. Please go ahead.
Oh, no.
Okay, I see you now. Okay, probably I can start with some question. So you give, I mean, what's this behind this interaction? As you mentioned, there is every cells express receptor for lipoprotein.
Yes.
What about EV? Do they have receptor for lipoprotein?
Yes, I think it depends. In our case, I think it would be really interesting, and definitely could go through some pre-existing proteomics data sets to find out and explore it in the future. But I also think it depends on the cell type as well. So for example, macrophages, they're foam cells. They are known to take lipid droplets and fatty acid droplets. So it would be more likely that they would also express the receptors. And so that is one of the mechanisms that we detailed in a previous kind of paper of how they could be interacting is through protein receptor interactions, but there could be a lot of different ways that these interactions are happening. And so whether it is just on the surface between two particles coming together through receptors and protein interactions is just one possible mechanism.
Okay, I guess this was a the very basic question because
No.
you know, it's something interact. We expect a receptor from both way. Any question from the audience? No. Okay, I will have a few more questions.
Yeah.
So, you know, every article related to extracellular vesicle, there is always the obstacle of standardization of purification, isolation of extracellular vesicle. What about the isolation of extracellular vesicle in your research? In this specific article, could affect also the interaction with lipoprotein?
Yes, definitely. I think, as you mentioned, standardization and even just the abundance of characterization tools and isolation tools that are available, definitely. So to break that question into two parts, so obviously it is a really big co-contaminant. So in our case, the cells that we use to derive extracellular vesicles, they were serum starved for 24 hours prior to extracellular vesicle collection. So we knew that they would have no lipoprotein contaminants that were present in the extracellular vesicles that we used in the assay itself. And we also validated all of our extracellular vesicles by cryo-TEM to show that there was the absence of lipoprotein particles that would then subsequently interfere. In terms of the second part of that question, there have been studies that have shown that different isolation techniques, whether that is what we used, which was tangential flow filtration, compared to SEC or density gradient ultracentrifugation, or change the protein corona of the extracellular vesicles. And so obviously, if we're thinking that it is a protein-mediated interaction, changes in the protein corona and the protein composition of extracellular vesicles would definitely influence the interactions. But that doesn't necessarily have to be a negative. If you isolate them one way and you see a binding, and you isolate them a different way and you don't see a binding, then you can actually probably compare them and see, well, what are the proteins that are changing? So it can be a pro, can be a con, but it is definitely something that will influence.
At some point, you show some data where you use plasma.
Yes.
What about the EV in there?
Yeah, yes, exactly. There would be extracellular vesicles that are already in there, and the images that we showed earlier on were just EVs and lipoproteins binding in just plasma samples. So it is something that is constantly happening. My way of thinking about it is that because it's constantly happening, when that is already accounted for in when we are doing the LAF assay, because they would have already been binding up until this point in the in the body, in the circulation, in the isolation. So then when we are adding extra on top, and obviously comparing it to a 100% control, so whatever is happening in the control is due to the EVs that are already in there. Those exogenous EVs that we are adding in are probably the ones mediating the effect rather than the ones that are already there.
Okay, thank you. So let's see if anybody from the audience. No. So I have one last question. It's more a general question.
No?
So LAF-ASA, I think, may have very huge potential in different direction. Diagnostic, also therapeutic, and also functionalization of the EV. Because, for example, myself at JotBody, mostly we work on the functionalization and drug loading. And we know that the corona itself is one of the huge obstacle to load any kind of drug. So where do you see the AFSA, the most relevant application? Diagnostic, therapeutic, functionalization? Tell me your point of view about it.
I think it just depends. All of it, right? Because in my opinion, it's really simple. So it is something that it can be automated. It's in a 96-well assay. It can be automated if you have the facilities. But even on its own, from firsthand experience, it doesn't take very long to set up, and it definitely doesn't take very long to run. So whatever application that you would like, I think it can definitely fit in. We work a lot in kind of biology and that area. So I think it looks really great in there to try and probe kind of different mechanisms of extracellular vesicle biology. But as you mentioned, the protein corona is a huge aspect, especially for therapeutic systems, because we want to make sure that will they all go and will they all target to the liver? Where will they try and go? And so why not, if you can do it in an hour, why not probe that interaction before you're running expensive, time-consuming in vivo clinical trials? So you can get this understanding and screen a wide range of compounds and different molecules to see, will they bind to lipoproteins? And is that actually something that you want? Where are you trying to target? What is your end goal? And go from there.
Can be also an essay to determine the level of purity of the standard that you are using somehow.
Potentially, yeah.
Okay. Yeah, I think that's I mean, there is a very huge room for application for this assay. So what's the next step?
I think for us is to just try and understand a bit more about extracellular vesicle biology, because we know that these interactions are happening, and I think the field is starting to kind of realize that they are probably not just contaminants, but they might also be important to extracellular vesicle biology. So now I'm very excited to maybe try and find out, well, how are they interacting? What are the mechanisms that are driving this interaction?
Yeah, I believe that everything is come from the, from the basic understanding. Then you can, okay.
And go
So
from there. Yes.
yes. Okay. So thank you again for your time. It was very, very interesting presentation. Good luck with your PhD, and I'm sure we will read more from you.
Definitely. Thank you. And thank you very much for the opportunity and for everyone for attending.
Thank you, everybody. See you next
Thank you.
time. Bye-bye.